Hello everybody, and thank you for watching my presentation. My name is John Walsh, and today we'll be talking about getting the upper hand on hypoxic stress. To understand the effects of hypoxic stress on the body, we first must understand the basics of the respiratory system. Now, the respiratory system is broken down into two different portions, the conducting portion and the respiratory portion. The conducting portion is the airway that leads to the respiratory portion. There is no gas exchange occurring, and the air is simply being prepared as it makes its way down to the respiratory portion. The respiratory portion is the area where gas exchange occurs. Anatomically, this is in the alveoli, which are made up of simple squamous epithelial cells, ideal for diffusion. Blood vessels around the alveoli provide blood which gets rid of its excess carbon dioxide and picks up essential oxygen from the air inside of the alveoli. From here, the circulatory system takes the blood back to the heart and then out to the rest of the body in order to oxygenate the tissues. Ideally, this occurs without a glitch, and homeostasis is maintained throughout the body. When this process does not occur flawlessly, then issues can arise. One of these issues is known as hypoxia. Hypoxia, very simply put, is a condition where tissues are deprived of oxygen. Now, there are many causes to hypoxia, and they typically involve interference of external respiration or internal respiration. External respiration is the process described in the last slide, how blood is losing carbon dioxide and picking up oxygen from the alveolus. Interfering with this process can cause the blood to not become optimally oxygenated from the lungs, meaning that there will be a shortage of oxygen in circulation of the body. Now, internal respiration involves the blood providing the tissues with oxygen and picking up excess carbon dioxide from these tissues. Interference with this process can cause tissues to not get enough oxygen from the blood and for the tissues to hold on to toxic levels of carbon dioxide. This brings us to the paper titled Mesenchymal Stem Cells Reduce Hypoxia-Induced Apoptosis in Alveolar Epithelial Cells by Modulating HIF and ROS Hypoxic Signaling by Olivier Bernard et al., published in American Journal of Physiology in November of 2017. In this paper, they are looking to determine if human mesenchymal stem cells have the potential to prevent apoptosis of alveolar epithelial cells undergoing hypoxic stress. It has been shown that mesenchymal stem cells work by secreting paracrine factors. So, in this study, they will be using human mesenchymal stem cell cultured medium as the treatment group. The mesenchymal stem cells were obtained through the iliac crest bone marrow of willing donors. Hypoxic conditions are simulated in a controlled chamber with 1.5% oxygen, and normoxic conditions were simulated in a controlled chamber with 21% oxygen. This is similar to the amount of oxygen you will see in the Earth's atmosphere. Now, alveolar epithelial cells were put under different conditions, and flow cytometry was used in order to count the amount of cells that were undergoing apoptosis. We will be starting with the figure on the left-hand side of the slide. Here, on the y-axis, is the percentage of cells undergoing apoptosis. And on the x-axis, there are different levels of treatment. Now, NX represents normoxic conditions, and HX represents hypoxic conditions. We are measuring the percentage of alveolar epithelial cells that are undergoing apoptosis after 24 hours in normoxic or hypoxic conditions in the presence or the absence of human mesenchymal stem cell cultured medium. Now, the white bar represents the alveolar cells without the cultured medium present, and the shaded bar represents the alveolar cells with the cultured medium present. It is first important to note that there is a significant increase between the white bars on the left-hand side and the right-hand side of the slide. This shows that there is a significant increase in alveolar epithelial cells undergoing apoptosis from the normoxic conditions to the hypoxic conditions, confirming that hypoxic conditions causes apoptosis of these alveolar epithelial cells. Next, it is important to compare the two treatments under hypoxic conditions. So we'll be looking at the right-hand side of this figure. We see a significant decrease in the percentage of cells undergoing apoptosis after 24 hours when the human mesenchymal stem cells are present. This shows a protective feature of the human mesenchymal stem cell cultured medium, that it decreases the amount of cells undergoing apoptosis when they are under hypoxic stress. Now, looking at the figure on the right-hand side of the screen, a similar conclusion can be reached. In this experiment, the researchers were measuring the level of caspase 3,7 activity on the y-axis under the same conditions on the x-axis. Caspase 3,7 is an essential apoptotic protein. The researchers concluded that the level of caspase 3 was significantly lower in hypoxic alveolar epithelial cells when the cultured medium was present rather than when it was absent. Again, this shows a protective feature of the human mesenchymal stem cell cultured medium, that it decreases the amount of cells undergoing apoptosis during hypoxic stress. This next figure here is very similar to the first figure that we looked at, in that they are again measuring the percentage of alveolar epithelial cells undergoing apoptosis on the y-axis and different levels of treatment on the x-axis. They are again comparing normoxic conditions on the left-hand side and hypoxic conditions on the right-hand side. They have the same treatment groups of human mesenchymal stem cell cultured medium and lack of the cultured medium. However, this time they have an added treatment group labeled heat. Now this heat treatment group came about by heating the mesenchymal stem cell cultured medium before the treatment of the alveolar epithelial cells for 24 hours. The treatments without the cultured medium compared to the treatments with the cultured medium render the same results, that there is a significant decrease in apoptosis when the cultured medium is present in the hypoxic conditions. However, when we look at the heat treatment group, we get a little bit of a surprising result, in that there is a significant increase between the human mesenchymal stem cell cultured medium and the heat treatment group. This shows that heating the cultured medium inhibits the protective effect of the human mesenchymal stem cell cultured medium. This led the researchers to believe that the cultured medium regulates apoptosis of the alveolar epithelial cells through the use of a proteic substance. So, to conclude, with this experiment, the researchers found out that human mesenchymal stem cell cultured medium has a protective effect on alveolar epithelial cells undergoing hypoxic stress, and that it is likely through the use of a proteic molecule that they offer this protective mechanism. Now, ideally with this information and through further research on the mechanism of protection, they will be able to develop treatments for patients with acute lung injury and limit the damage caused by hypoxic stress. The discussion questions that I prepared today are as follows. What conditions or illnesses can cause hypoxia in tissues of the body, and why? And why is hypoxia so harmful to cells? Thank you all for listening, and I'll see you in class.